Hello, students. This is our second week topic of analytical techniques module. Today we are going to talk to you about microscopy. I hope you will find this topic very interesting because it is actually used in different fields of research and practice. Okay, first let me tell you briefly what the topic is about. First, we are going to understand what the microscopy is. And the first part will be about light microscopy. And then we will talk about electron microscopy that uses electrons. And then we will talk about scanning probe microscopy. And then we will have the summary. Okay. The fundamental problem of microscopy comes from the resolution. You know that our eye can see the very short wavelength, very short range of the electromagnetic waves ranging from 400 to 700 nanometers from the violet to red colored electromagnetic waves. This is the only colors that our eyes, by using protein termed rhodopsin, can identify. So this is the main reason that why we can't just look closer. And 2 points of light close together appearing as one blurred spot. Okay, the resolution limit is the main point. Resolution is the minimum distance two objects can be apart and still can be seen as separate. On the basis all of these processes, there are the properties. There is the properties of electromagnetic waves. So, in order to understand this topic, I strongly recommend you to watch some videos on YouTube about the electromagnetic waves and how the microscopes work. Okay, we cannot resolve details smaller than roughly half the wavelength we are using to look at them. As we said that this is the wavelength range that our eyes can identify. Okay, now we are going to talk about microscopy spectrum that can be compared to the abilities of the human eye. As we notice here that the human eye at maximum can identify 0.1 millimeter objects, and the smallest object that our eye can identify is 100 micrometers. And this is even the smallest capacity of light microscopes, and light microscopes can identify until 200 nanometer size, from 200 nanometers, 100 nanometers, until let's say 200 micrometers can be visualized by using light microscopes. And electron microscopes have higher, let's say, capacities to turn those properties into the color images that we can see. And electron microscopes can visualize the objectives ranging from 0.1 nanometers to 100 micrometers, as shown here. So by using electron microscopes, we can see the sizes of cells, bacteria, virus. We can see the DNA molecule. We can see proteins, and some small molecules like glucose can also be seen, and some higher higher sized atoms can also be detected by using electron microscopes. Okay, as the smaller sizes are required to be detected by electrons, okay, it will be more difficult. This is about electron microscopes, and scanning probe microscopes have even higher ability to see in the smaller, let's say, objects in atomic size level. Okay, the conclusion is the smaller the object, the smaller the wavelength is required. This is the reason. Okay, since the light has higher, let's say, wavelength, so its ability and is okay here. And since the electrons wavelength has the ability until here, we can use electrons to see the atoms. This is about the physical properties. In order to understand the electron microscopes and other types of microscopes, we need to go deeper into the physics of electromagnetic waves. Next. Okay, and now we are going to understand some light microscopes. And when we use some light microscopes, okay, sometimes we can see, we can obtain this kind of images that are not, let's say, very clear. So by enhancing the contrast, we can obtain this kind of figures, this kind of images, which can tell us more about the properties of small objects. They can be biopolymers, they can be small molecular, smaller molecules, higher molecular compounds. And it converts subtle differences in the thickness and refractive index of the cell, which alter phase light into different brightness. I express my apologies. I'm sorry. This is about the cell, actually, not biopolymers. I apologize. This is about light microscopy. Okay, and light microscopy uses polarized light and prisms to create a 3D shadow cast relief image. So light microscopy can be used to see the cells, bacteria, and some higher size virus as well. But usually, virus can be seen by using electron microscopes. Okay, next. And fluorescence microscopy can also be effectively used to see the cells, even cell organelles. Here we can see different organelles of the cells that could be shown by using fluorescence microscopy. Fluorescence microscopy is used to paint the cellular world, subcellular world as well. A colorful fluorescence micrograph of a cell with different organelles can be obtained in different colors. And the principle comes from the molecules that have fluorophore groups, which absorb high energy, short wavelength light and emit lower energy, longer wavelengths. This is the fluorescence. So if we provide some molecules having fluorescence property, so then we can identify the molecules we are administering can have affinity and can attach to those molecules. And the techniques used here are tag-specific proteins with fluorescent dyes. One of the most used in biomedical research is green fluorescent protein. As I said, okay, it's applied to see the location of subcellular organelles. Here we can see plasma membrane, mitochondria, Golgi apparatus, endosomes, peroxisomes, lysosome, nucleolus, nuclear envelope, nucleus. So fluorescence is one of the approaches that we can use to see subcellular organelles, subcellular compartments. Okay, next. And this is about confocal microscopy. Confocal microscopy is used to develop 3D structures of cells. This is that we can obtain fluorescence microscopy by using fluorescence microscopy. This is about 2D structures. And here we can see 3D structures. The 3D structures tells more about the properties of cells, of microorganisms. So confocal microscopy can also be effectively used to study and to study in research and in biomedical research. Okay, and it helps us to develop diagrams that shows a laser focused. I mean, it shows a laser focused on one plane of a thick panel. Of a thick sample, I'm sorry, a pinhole blocks out of focus light, and confocal microscopy uses a laser and a pinhole to eliminate out of focus light and scans the samples point by point to build an image, so that it develops a high resolution 3D images. Let me tell you that laser is the abbreviation of light amplification by stimulated emission of radiation. It can have different types of ranges of electromagnetic waves. We often use the red-colored laser in biomedical research. Yeah. So this is about the confocal microscopy. Next. And some confocal microscopy images may not be clear enough, so we can use even better resolution, which is called super resolution microscopy. Here we can see one of the methods of super resolution microscopy, STED. Compared to confocal microscopy, here we can see much higher resolution of the object. STED stands for stimulated emission depletion microscopy. Besides, there are some other approaches of super resolution microscopy, such as photoactivated localization microscopy or stochastic optical reconstruction microscopy. And they develop images to compare a conventional fluorescence image of a microtubule vs a super resolution image and show the hollow tube structure as shown here. And the concept is techniques that circumvent the 200 nanometer diffraction limit. I think for developing this kind of technique, Nobel Prize was given in 2014. Okay, STED uses a donut-shaped laser to show the fluorescent spot, and the resolution it can be achieved to 10 to 20 nanometers. And this is about electron microscopy. Now we are going to turn into electron microscopy. Electron microscopy used electrons from photons to electrons. It is used to see smaller details, use a smaller wavelength. Electrons accelerated by high voltage have wavelengths thousands of times shorter than light, so they can go deeper into the objectives, which enable us to see inside the biomolecules, biopolymers and low molecular compounds as well. And instead of glass lenses, electron microscopy uses electromagnetic lenses to focus the electrons beam. And it must be performed in a vacuum. So this is one of the pointers. And here I provided one video file from one link to YouTube. In a video file, you can get acquainted with how the transmission electron microscopy, in general, electron microscopy works. This is about the mechanism of electron microscopy. And this is about transmission electron microscopy. Actually, there are two main types of electron microscopy. And the first one is transmission electron microscopy. The second one is scanning electron microscopy. Transmission electron microscopy have this kind of complex mechanism. Okay, electrons are directed from one place to another, and at the end, the changes in the electrons, let's say, absorption and reflection are turned into the color figure that we can see. And transmission electron microscopy is used to see internal structure. It does not develop a 3D structure. For that, electrons pass through an ultra-thin section of specimen, and denser areas stained with heavy metals like uranium, lead, and scatter more electrons appearing dark. And high-resolution image of a mitochondrion showing the details per state can be obtained, and atomic lattice resolution can be achieved to 0.1 nanometer. And this is about scanning microscopy. And scanning microscopy mechanism is also quite complex, and it develops a kind of 3D structure, let's say. It is used to see surface topography, and an iconic scanning electron microscopy image of a fly's eye or pollen grain. A focused beam of electrons scans across the surface of the sample and detects secondary electrons knocked loose from the sample surface, resulting in a stunning 3D structure image of the surface texture, and the resolution can be approximately to 1 to 5 nanometers, less than scanning electron microscopy, but greater depth of field. And here we can see the differences in the transmission electron microscopy and scanning electron microscopy. Again, transmission electron microscopy develops a 2D structure, two-dimensional, and scanning electron microscopy develops a three-dimensional image. Here we can see the TEM develops internal structure, SEM develops a 3D structure, and TEM extremely thin slices for sample preparation, and SEM bulk coated with conductive metal, and electrons are transmitted through sample, and SEM electron scanned across the surface, and maximum resolution, as we said, differ. SEM have not as effective as TEM in resolution, in terms of resolution, but it develops 3D structure. And analogy, like an X-ray, can develop this kind of figures, and like a magnifying glass, we can see this kind of objectives by using SEM. And here we can see the comparison of optical microscope image of nanofiber and those that were obtained by using scanning electron microscope. As we see that by using scanning electron microscopes, we can see the 3D structure in the same figure. But by using optical microscope, we are unable to see different layers. So different slices can be seen by changing the distance of the lens. So then slice by slice, we can see different layers of the object by using optical microscope. And scanning electron microscopy enables us to see to obtain this kind of image. Next, and the challenges of electron microscopy are given here. It's not as simple as using light microscopes, and it should be carried out in vacuum. And fixation is also kind of fine art, chemical preservative to prevent decay. And the sample should be dehydrated, replacing water with a solvent, and infiltrating with resin plastic to harden the sample is required for TEM. And slicing with a diamond knife can be required, and staining can be required in some cases. Okay, staining can also be used for light microscopy as well. And here, this staining stands for using heavy metals to provide contrast. And coating can be required for scanning electron microscopy. And spar coating with gold or platinum can be required. And scanning probe microscopy is another type of microscopes. This is about the field. Atomic force microscopy is one of the types that uses the kind of field, which uses different approach compared to the first two ones. Let's say it moves beyond the light and electrons. And the concept is about instead of looking at the sample with waves, here it's used kind of feel it with a physical probe approach, which is about using light and electrons. And it can operate in air, vacuum, or even a liquid. It does not require a vacuum condition that are used for electron microscopes. And two main types exist, as I said, atomic force microscopes and scanning tunneling microscope. Here we can see that atomic force microscopy is one of the types. It uses a cantilever and a laser, a sharp tip on a flexible cantilever touching the surface is used there. A laser reflects off the cantilever into a detector, and the principle is a sharp tip scans across the surface. Interatomic forces like van der Waals interactions between the tip and atoms deflect the cantilever. This is, as I said, is a more differing approach from the light microscopy. Let me tell you again, light microscopy uses light, electron microscopy uses the flow of electrons, and the atomic force microscopy uses interatomic forces between the tip and atoms that deflect the cantilever. And then a laser measures these tiny deflections. A computer then maps the topography by using special tools. So then a 3D height map of the surface down to the atomic level can be obtained, it can be achieved. Here is the example of atomic force microscope by going to this website on ResearchGate. You can see the you can read the whole paper, or you can see other figures of that photo. Here we can see a 3D structure of one example. Examples of results of scanning cells. I don't know what kind of cell it is. So here we can see the 3D structure of one type of cell. Okay, next, scanning another type of microscopy. This is scanning tunneling microscopy. It also used to see electron. It is used to see the electron clouds, the iconic quantum coral image. And the principle is that it is a conductive tip. A conductive tip is brought extremely close to a conductive surface, a voltage is applied, electrons tunnel across the gap. The tunneling current is incredibly sensitive to distance, and the requirement is samples must be conductive. If the electrons cannot be brought, let's say, and cannot go across the gap, so then we cannot obtain some kind of images like this. Here is the STM topography, scanning tunneling microscopy topography image of a 5 nanometer large void in a topological insulator film, potential dipole developing around the nanoscale void due to the current flowing. Okay, next. And this is about the summary, how to choose a microscope. Okay, if the first question is the sample alive? Okay, so if the answer is yes, we can use light microscopy. We can enhance phase contrast. We can use DIC. We can use fluorescence. And we can obtain different figures of cells, and in some cases by using fluorescence microscopy we can see subcellular compartments, organelles. And if the answer is no, okay, so then another question can be given, whether we need to see inside or the surface. If we want to see inside, okay, we can use TEM. Let me tell you that TEM, SEM, and others can also be used for live cells. By non-live objects, we mean smaller objects, okay? Because these cells are the smallest unit of the life. So if we want to see the structures, let's say two-dimensional structure of subcellular compartments, we can use transmission electron microscopy. If we want to develop a 3D structures in a micro size level, so then we can use scanning electron microscopy, or for seeing some objectives in a nanometer scale, we can use atomic force microscopy, or just as we talk, scanning tunneling microscopy. So these are about microscopy. I strongly recommend you to watch some videos if you want to go deeper into understanding how those microscopes work, and determine the objectives. So then you can have a better imagination about those methods. And on Thursday, we will talk to you about microscopy more in our seminar. That's all for now. Thank you for your attention.